Okay, for this lecture, lecture 4.2a, we're going to be looking at eukaryotic cell organelles. Okay, I'm not going to spend too much time on this. You have seen this. You have had this lectured to you since you were in junior high, high school, and hopefully either in, concepts of biology or intro to cell and molecular. You've seen all these, so I don't have to spend too much time. We're going to be looking at, quickly discussing some of the important key features. We're going to be looking at some of the things that are important to know. When you look at the external structures on eukaryotic cells, one of the things that we notice are appendages that are used for locomotion. Two types of appendages can be used for locomotion here, cilia and flagella. Now keep in mind, the flagella here that we're talking about, it's not like the ones that we talked about in the last module with bacteria. Bacteria had this thing that was outside the cell that had a motor that spun it around like it was, a jump rope. Here we're going to see that flagella and the cilia are actually internal structures that just kind of push the plasma membrane, the cytoplasmic membrane, out. As you see on the figure next to me here, this series of circles is actually when if you were to take a cilia or flagella and to cut it and look at it end on. So it's an end on cut. You see that it's a series of rings. Well, it's not actually rings, they're tubes. It's the microtubules that form this formation. These structures give the cilia and the flagella their rigidity, yet allow them some movement to be able to bend. Cilia move like this. The flagella, it's going to be more of, think of it when you're swimming in the pool and how you flutter kick your legs, except it's going to be a lot faster. What you have are those tubules. You see there's 9 pairs in the circle, 2 in the middle. The 9 pairs just kind of rub up against each other. And as they're moving, rubbing on each other, they're causing themselves to bend back and forth. How important is this for you to know? Well, it's kind of important because it's one of the pillars of evolution. All eukaryotic cells that have cilia or have flagella use that structure. That structure, that 9 plus 2, is in all eukaryotic cells. Okay, from some of the oldest single cellular microorganisms up to us here in the now. Some of the other external structures you may see in some eukaryotic microorganisms. An outer protective coating called the glycocalyx. Yeah, we talked about this back with bacteria. We're going to see it here again with some protozoa, maybe in some fungi. Again, it's an outer protective coating, protect from water loss, protect from chemicals, protect from larger predators. For many of our microorganisms we're going to talk here, soft, squishy. Most of us eukaryotes are soft and squishy. What you're going to see, though, when the microorganisms we're going to be talking about, the fungi, are going to have a hard outer cell wall. Gives them structure, quite similar to what we saw with plants. Not the way they're made, but their purpose. Protect the cell. Allow for the tissues that the cells form to have some three-dimensional shape that can't be changed due to osmotic pressures. So when you look at a fungal cell wall, you'll see there's the cell membrane. Right outside of that, chitin. Outside of that, a glycoprotein layer that is attached to some form of a glycocalyx, that protective outer coating. You can see this with some fungi that are single cell, like the yeast cells. You can see it with some protozoa. Now, when it comes to the cell membrane, for all intents and purposes, In the eukaryotes, you're going to see it is a lipid bilayer. Well, they're all lipid bilayers, phospholipids, but where it changes is what's in there. What form of phospholipids? You could take the phospholipids, you have the tails, the glycerol, the phosphate head, and then on top of that you can add amino acids. You could add sugars or a mixture of the both so that you get different flavors of phospholipids. One of the things you're going to see with eukaryotic cell membranes is that interspersed throughout are going to be different types of sterols and steroids. These sterols or these steroids act as spacers, kind of push the phospholipids away from each other so that way they have room to move, because if they're packed in, nobody moves, and you lose that fluid of the fluid mosaic.